Alright, welcome back. In this video, we're going to go over classes of amino acids and special properties. I made a lifetime total of $124.56. We broke $100. That's amazing. Thank you so much for the support, guys. Okay, so I've got a lot of comments recently from my subscribers asking if I could make a video on the different classes of amino acids and tricks to remember which amino acid goes under what class. For example, the non-polar, not aromatic amino acid group, the polar uncharged amino acids, the non-polar aromatic amino acids, positively charged R groups, and negatively charged R groups. So I have a trick that you can use to remember all of them and what amino acids would fall under each group by just looking at them. I promise it's going to be really helpful. In addition, I want to go over the special properties of each individual amino acid because I've been seeing your professors are asking you questions for example, what amino acid is involved in N-linked glycosylation, or what amino acid is involved in phosphorylation, and you have to pick the amino acid or whatever, multiple choice. So I'm going to go over them. So I promise you that all of these here are all of the tricks or all of the special properties I'm going to say would be something you would probably see on an exam. So it's going to be very helpful. Okay, I want to make a little disclaimer here. This video is going to be very rated R. And the reason I'm saying this is because there are many ways to remember the amino acids via mnemonics. Most of the common mnemonics don't really help me. So I came up with very dirty ones to help me. Now, they may help you, they may not. If they don't help you, then just ignore them. If they do help you, then great. But just don't get offended. Just warning you. Okay. Let's start with the non-polar aliphatic, which means not aromatic, amino acid groups. The trick for these is all the amino acids you'll see here in this group have no electronegative atoms. There are no oxygens, and there are no nitrogens anywhere in the R chains. We're only talking about the R chain, right? Not the amino group here or the carboxylate group here. I'm just talking about the R groups, which are in red. So no nitrogens and no oxygens. And sulfur can be deemed electronegative, but there is a certain very special circumstance with methionine, which we're going to go over. So let's start with glycine. Glycine is the most basic amino acid. It only has a hydrogen in the R group. Glycine with a G, or gly, is involved in very flexible bonding and folding with amino acids and protein structure. The reason is because the amino acid is so small, it's able to bend, and it's found at beta turns and loops when proteins are trying to fold. Glycine is also found in the brain as a neurotransmitter. So neurotransmitters are basically chemicals that neurons use to communicate within each other. It is the second most common inhibitory transmitter. So in other words, it makes neurons not fire. The most prominent one is GABA, but glycine is the second one. Alanine is the next amino acid, and it's boring. It really doesn't do anything except hydrophobic bonding within the core of amino acids. So the hydrogen from glycine is replaced with a methyl group, and that becomes alanine, A for short, or ALA. Now, all of these amino acids here are located inside the core of proteins, the hydrophobic core. They do not like to be on the outside. They like to be in the inside. So non-polar, not aromatic amino acids are located inside the proteins, inside the core, and they all interact with each other. Valine is the first interesting amino acid here because it's the first branched chain amino acid. We call this a branched chain amino acid because it branches out from a CH to two methyl groups. Val, or V for short, is found in the composition of hemoglobin. So remember that you probably learned this in biology or molecular biology, or learning it now. DNA makes RNA, RNA to proteins. The RNA then has the codons, right? We care about. After RNA, we have the codons. AUG is the start codon and codes for methionine. So all proteins out there start with methionine in the amino acid sequence. Now, that doesn't say that methionine is still there at the end when the protein is made. It could be chopped off. In the case with hemoglobin, very interesting, with hemoglobin, the structure of hemoglobin always starts with valine. It does not start with methionine. It starts with valine. But when you make hemoglobin, when you actually form the amino acid sequence, and then we get the folding, to the folding, we get to the part of the folding, it starts with methionine. So when you're making hemoglobin, we have to start with methionine because that's the start codon. AUG is the start codon, and it begins synthesizing methionine and then whatever the rest of the amino acids are. Well, whenever hemoglobin is done forming and all the folding is done, the methionine gets chopped off, and the very next amino acid in the sequence is always valine. So, a little interesting fact there. Leucine is the next one. It's also another branch chain amino acid, CH2, CH in 2 methyl groups. We're just adding a CH2 from valine. Leucine, L or Lu, is interesting because there's a very unique motif. There's a pattern. Now, that doesn't happen with all proteins, but some proteins, what scientists have noticed, chemists have noticed, that every 7 amino acids tends to be leucine. So you could have leucine, then blah, 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 6 more amino acids, and the next 7th amino acid would be leucine. So 7, 14, 21, and so on. Very interesting, right? Peculiar that every 7th amino acid tends to be leucine. Okay, methionine. Like I said, the start codon is AUG and codes from methionine. So all amino acids start with methionine. All proteins start with methionine. So when we're building a protein, all of them must start with methionine. This is not to say at the end, when the protein is done and done folding, it'll still have methionine as a start. It will not. Most of the time it gets cut off. But to actually start building a protein, methionine is always first. And methionine has the sulfur group. You'll notice that yes, sulfur can be electronegative, but it's surrounded by so many carbons, hydrocarbons, it basically is nonpolar. The electronegativity of sulfur basically just gets canceled out because it's just so much hydrocarbons around it, making it so nonpolar. So that's why it doesn't really do anything. Isoleucine is next. Isoleucine, nothing really fancy going on. It's just hydrophobic like all the other amino acids in this group, and it basically strongly bonds inside the core of proteins with other nonpolar amino acids. So nothing really going on here. Just CH, methyl on the side here, and CH2 and a CH3 there. What gets interesting now is proline. Proline is not a cyclic amino acid. It's not aromatic. The reason is because to be deemed aromatic, it must be part of a conjugated pi system. If you have not taken organic chemistry 2 or organic chemistry B or whatever you say in your school, what conjugated pi systems really mean is that there has to be a double bond every other bond. There are no double bonds at all in proline. So we cannot say it's an aromatic amino acid. We're not allowed to say it. What's interesting about proline is that it has a secondary amine. And due to this pentagon-looking shape it has, it's very rigid. And it's found a lot at bends and turns of amino acids, and it causes something called kinks. So it causes like bumps or ridges in the protein structure due to its box look-like structure. Now, the mnemonic for the nonpolar aliphatic, not aromatic amino acids is greedy assholes very likely impregnate many people. Don't ask me, just go with it. If it works, it works. It doesn't, whatever. Okay. Now let's do the polar uncharged amino acids. Here's a trick. It has electronegative atoms. Can you believe it? This one, no electronegative atoms. This one, all electronegative atoms. Da, 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 da. Okay. The first one is serine, S or SIR. Serine is very important as a phosphorylation site. It is used as a phosphorylation site. This is very important for the chymotrypsin mechanism if you happen to learn about the chymotrypsin mechanism. Most biochemistry courses, you do learn it. You may not, but I'm just telling you this is very important for the chymotrypsin mechanism and other enzyme reactions out there. There is something called O-linked glycosylation. I'm going to go over it when we talk about asparagine down below, so keep that in mind. There's also threonine. Threonine also does O-linked or oxygen-linked glycosylation. Also, phosphorylation sites. So they're basically the same. Serine and threonine are very alike. They look different, but very alike. Now, with threonine, there is another amino acid called theanine. Theanine is not part of the human body. It is made by plants. So don't get confused between threonine and theanine. There's no R in theanine, which is in plants. You may have heard of L-theanine. There's a very interesting reaction that can happen with caffeine. This is just outside information, just if you're interested. If you happen to get caffeine jitters or you don't like the spike you feel with caffeine and then just like wears off like after an hour, you can take theanine, L-theanine with caffeine. What happens is when you take theanine and caffeine together, you do not get the jitters, nor do you feel that caffeine spike. Instead, you get that attention longevity over hours, not like 1 hour. So like if you were to take a cup of coffee and you would feel that effects for like an hour, right? Or maybe 2 hours. And then you just feel like the caffeine just wears out and you feel awful. If you combine theanine, L-theanine, and caffeine together, you won't feel that. The caffeine effects will actually last for 3 to 4 or 5 hours. Prolonged. This is actually what physicians do when they have to do a long shift, or when you go to a club, people also do this to stay awake. It's kind of interesting, very interesting reaction. Cysteine is probably one of the most important, they're all important amino acids, but this is the very unique one because of the thiol group here. Cysteine is able to do something called disulfide bonds. Basically, the sulfurs can bond with each other. So cysteine amino acids can bond with other cysteine amino acids. This is critical for tertiary and quaternary structure. That being said, I said sulfur and sulfur are disulfide bonds. Does that mean cysteine can bond with methionine because it has a sulfur? The answer is no, because it is essential for disulfide bonds to have that hydrogen here. In order for this to actually work, you need a hydrogen. And cysteine has it, methionine does not. There's no hydrogen here, it doesn't exist, so it cannot do it. In addition, if you happen to have very wavy or curly hair, you have a lot of cysteine in your hair. You have a lot of disulfide bonds. Just a little interesting. It causes curls and waves in your hairs. Okay, let's talk about asparagine. Asparagine, or acin, or N, can do something called N-linked glycosylation. Okay, so let me explain what this is, because it's very important for you to know. Glycosylation is when this amino acid can bind with a sugar. This can only happen when asparagine is first bonded with another amino acid. It can be any amino acid that is not proline. And that other amino acid must be bonded with serine or threonine. Here. When this happens, when you have this 3 set up amino acid here, a sugar has the ability to bind where the nitrogen is, the amino group is. This is called N-linked glycosylation, nitrogen-linked glycosylation. Now, it can go the other way. The sugar can actually bond to serine or threonine at the oxygen site here. So we can have the opposite. We can have asparagine alone here, bonded with whatever amino acid that's not proline, serine threonine, and then the serine threonine can have a sugar bonded to it at the O site, the oxygen site. So this is why it's called O-linked glycosylation. Why is this important? This is important because proteins are made in the rough ER. For proteins to be formed correctly and folded correctly, it needs something called chaperone proteins. Chaperone proteins are basically there to help make sure and ensure the proteins are folded correctly and there's no misfolding. It's like an assister. It's kind of like a personal trainer when you go to a gym. You may go to the gym for the first time, you may make mistakes. We have a personal trainer there to help you out to make sure you don't make those mistakes. That's what's happening. So what this does is this whole complex here with the sugar and 3 amino acids, it's a signaling molecule for the chaperone proteins to come over and basically watch the protein being folded to make sure it works properly. So all it is a signaling molecule to signal for the chaperone protein to come along and watch the protein fold correctly and make sure nothing happens. Otherwise, it'll fix it. Okay, glutamine is next. Glutamine is not the same thing as glutamate. They're two different things. What's very interesting about glutamine, besides it has the abbreviation as a Q for who knows why, it has the ability to donate an amide group here. And it also can perform very strong hydrogen bonding with other molecules. So that's it. Nothing too special with glutamine. Now, the abbreviation that I like to use is sexy, thick, young couples always grind. Okay, so let's go over the nonpolar aromatic amino acids. The trick is all rings, so they're all rings, with no electronegative atoms besides tyrosine. I know that doesn't really help you because there's only 3 here, but you just gotta remember tyrosine. Tyrosine is my favorite amino acid, by the way. I'll tell you why in a second. So let's do phenylalanine, or F for fuck. What's interesting is only a CH2 in a benzene ring. It absorbs UV light at 260 to 280, so we can easily detect it. That's basically it. Now, tyrosine is the exact same thing, but with an OH group at the very bottom. This is the precursor to dopamine. Dopamine is a reward neurotransmitter in your brain. So whenever you feel like a rewarding feeling, for example, you won the lottery or you got an A on your exam, that rewarding feeling you feel, that great feeling you feel, is dopamine, which is also the target of cocaine. So that's why. So tyrosine is a precursor to it. So if you don't have tyrosine, you're not going to make dopamine, and you won't feel great. You're going to feel depressed. So tyrosine is abbreviated as Y or TEAR. Tryptophan is the precursor to serotonin, the mood neurotransmitter. So this is your overall mood. Do you feel happy? Do you feel sad? That's what serotonin is. It's also key in phosphorylation, so it helps with phosphorylation. Now, tryptophan is very, I'm going to be honest, kind of hard to memorize because it looks so awful. And you know it's awful because it has the abbreviation W for no reason. And TRIP is the shorthand, three-letter abbreviation for tryptophan. There was an experiment a long time ago that was done. What they did is they took a group of people and they degraded all the tryptophan storages in their body. And they wanted to see what happens. And boy, did they get depressed. They got so depressed that they had to end the experiment early, if I'm not mistaken, because they felt really suicidal. So make sure you have tryptophan in your body, otherwise you're not going to be that good. Okay. Oh, and the abbreviation, the mnemonic is fuck your wife. Now let's do the positively charged R groups. Here is the trick. Only the nitrogens carry the charge. So you're going to see nitrogens in all the positively charged R groups. What's easy about this to remember is the positively charged R groups have nitrogens or amino groups. The negatively charged only has carboxylate ions. So no nitrogens whatsoever. They are all basic and they're all polar. So here's just basically a chain of CH2s in the amino group at the end. That's lysine. So lysine, arginine, histidine, Aspartate and glutamate can all form salt bridges. What salt bridges are is basically a bonding between a negative amino acid and a positively charged amino acid. So when a lysine bonds with aspartate or glutamate, or arginine binds with these two, or histidine binds with these two, that's a salt bridge. Easy way to remember this is sodium chloride is salt, right? Sodium chloride is salt. Well, sodium is positively charged, chloride is negatively charged. They come together as a salt, right? So that's how you remember. Okay. Lysine is involved in acetylation and ubiquination. That's all you really need to know there. Arginine can form salt bridges, and it's also found in the backbone of DNA. Not really important either. Histidine now is the really unusual one. You will notice that histidine has no positive charge on it. I drew it, no positive charge, besides, here, but that doesn't count. Only talking about the R groups. Why is this? The reason is histidine fluctuates in charge at physiological pH. It's not stable whatsoever with the charges. In fact, at physiological pH, histidine has a 15% charge that there's going to be a charge on there, a positive charge. So we just say it's part of the positively charged R groups. Due to this unique characteristic, it's an amazing proton donor and acceptor because it's losing that proton, it's gaining it all the time. So it's amazing at it. And it's so common in enzyme activation sites to help reactions go forward and go faster. Lastly, let's do the negatively charged R groups. The trick is the oxygen holds the negative charge. There are no amino groups here, only carboxylate. And that's what holds the charges here, both of them. They're both acidic and both polar. Okay, aspartate. Involved in many enzymatic active sites. That's basically it. Glutamate now is the most common excitatory neurotransmitter in our brain. So neurotransmitters, once again, basically are chemical messengers. This is the most common excitatory messenger. So basically activates neurons. This does not mean that you're more alert or more awake, right? That's not what it means. It just means it's activating a neuron, but that neuron could do something else. It could make you depressed. So that doesn't really mean it's making you happy or anything. It's just exciting the next neuron in the pathway. And that's glutamate. And that's it. If you found this video, please like and subscribe. Oh, wait. I almost forgot. The mnemonic for positively charged R groups is kinky rough handcuffs. And the negatively charged one is deep entry. So yeah, okay, we're done. If you found this video helpful, please like and subscribe. Until next time, later.